Good morning again. So to functional connectivity and dynamical functional connectivity. My name is Gijs Plomp, and I'm with the Perceptual Networks group in Fribourg. And let me just show you what the plan is for today. We will talk a little bit conceptually about what functional connectivity is. And then we'll dive a little bit into more on the relationship between, we dive into the relationship between functional connectivity and physiology. So that's basically the main point I want to get across today. What we've been trying to do in our work and in some parts of the Synergia is to try to bring functional connectivity closer to physiology. So basically trying to take into account rhythmic aspects, trying to take into account fast dynamics, and try to take into account directed interactions. Then I'll briefly conceptually introduce the concept of Granger causality and one particular instance of it, the partial directed coherence. I'll talk about a few limitations that are particular to EEG and MEG when we do this type of functional connectivity analysis, and talk a little bit about applications. So this talk is going to be a little bit conceptual. There won't be very much formulas in there. But it's if you like, it prepares the ground basically for the lecture that comes afterwards by David, and the tutorial that is this afternoon given by Maria. So with that, let's get to the main dish, which is functional connectivity. Well, functional connectivity, as you've already heard in a few talks, is statistical dependencies between recorded signals. Well, this is, of course, a rather broad concept, and it can mean different things. But in general, what it means is that we record signals from a brain. Of course, we record multiple signals because we're interested in how different brain areas interact, basically. And for each of these recording sites, we have time series. They just go like this in time, and we devise some measure of the similarity or the relationships between those time series. And when we say there is such a relationship, then we draw a line between those 2 points. That's in the broadest conceptual sense what functional connectivity does. Now, so essentially this indicates how activity in one area relates to activity in another area. And the advantage of that is that we can move to sort of a network perspective on brain function. Instead of looking at how activity in single areas changes between conditions or changes over time, we look now at how certain properties of networks or certain interactions between areas, or say the driving force of one particular area, changes in time. So that is in a nutshell. Yeah, that is in a nutshell what functional connectivity is very broadly. Now, we've already heard we've already heard in these lectures, and obviously also in our lives, about correlation coefficient. This is the simplest form of functional connectivity. So here I show two simulated signals. It's just a sinoid with a little bit of noise. If we have, for example, a signal like this from area 1 and a signal like this from area 2, there will be a very high correlation between them. So this is almost a perfect match, 0.92. That is the simplest measure of functional connectivity. Simple is often good, not always, but particularly in fMRI, this measure is used as the default measure in functional connectivity and network type of analysis. Here are some examples of resting state fMRI that you've already seen, I think, in one of a few of the talks. But here, generally, the idea is, say, if you take a seed voxel and you correlate that with activity all over the brain, there is a pattern that emerges, and these patterns are meaningful in the sense that they're very consistent and help you sometimes even distinguish between patient populations and normal healthy controls, etc. So this is a very useful way of looking at the brain in fMRI, the correlation. However, when we're interested in doing functional connectivity using MEG or EEG, there is one problem, and that is that MEG signals, they show rhythms and oscillations. I think you've seen already in Serge's talk, Serge Plum's talk, that Berger, when he started recording the EEG with just two simple points on the head, he already saw one striking thing is that, you know, things go like this, they oscillate in the alpha rhythm in his case. And actually, when we record an EEG, there are many different rhythms and frequencies in there simultaneously. Now, that means that the signals look a little bit more like this. So here we have, the signal that I from the previous slide, the one before that, a simple oscillator. And here we have the same oscillator, but there's a lower frequency overlaid with it. Now, if you calculate the correlation between these two, the correlation will have dropped from 0.9 to 0.4. So does that really mean that there's no relationship between those two signals anymore? Not really, because in the reality that is underneath, so if we, which we see if we do the Fourier transform of this signal, we see that this signal has two peaks in the two peaks here and here in the frequency domain, one at 10 hertz, say, and one at 1 hertz. Whereas the other signal has only a peak at 10 hertz. And now if we calculate a correlation for each of these frequencies, we see that there is a strong dependency, a strong functional connectivity between those two signals only at 10 hertz, but not at 1 hertz. So this frequency specificity is the reason why the coherence is in EEG and MEG usually a traditional method of choice. So it's a correlation value at each temporal frequency, and is the standard methods, the standard method in MEG studies. So we'd like to take into account these rhythms, but there's another aspect about brains. If we want to understand the brain and we want to understand it as a network of working together areas, we have to take into account the fact that brains are extremely fast. I mean, and this is so obvious that we don't realize it in day-to-day life. I mean, of course, we can marvel at these Olympic players, at how fast they hit the balls, or at how well somebody can drive at high speeds, or how agile certain cows in Switzerland are, or how a frog leaps. But you know, even when you listen to me talking now, or you type something, you do multiple things per second, and all these things that you do require coordinated activity throughout different brain areas. So this is an essential property of how brains function. So how can we study these fast responses? Well, obviously, what we do often in the lab is present stimuli repeatedly, and we create an evoked response. This is an example of an evoked response in the EEG, visual evoked response. And it's just to illustrate that when we present a visual stimulus, that already after 70 milliseconds, you know, there's this big distributed pattern of activity over the head that again within another 100 milliseconds changes into something completely different. So that means that within a second, so and here I've plotted not even half a second, you know, several different states can be discovered. And if you want to understand, for example, visual processing or auditory processing or tactile processing, we're confronted with the problem that these states alternate very quickly in time. So we have really a need to work with dynamic models, models that can deal with these types of non-stationarities, these type of fast dynamics music. So after rhythms, dynamics are important to account for when we want to do functional connectivity analysis using EEG and MEG. A third one, and we've heard this already a little bit in Jonathan's talk, and also the question about Granger causality, is that when we look at the physiology, we have to, and we take interactions between brain areas really as something physiological, then we have to acknowledge that neural interactions are inherently directed interactions. Basically, in area 1, in a cartoonish fashion, you know, there are neurons that generate an action potential that travels through the axon, comes into another area, and there changes the spike likelihood of other in that particular area. So interactions in the brain are always directed interactions. So it would be interesting if we can have a directed network, basically, of functional interactions. So to sum up, in the work that we've been doing, we emphasize three key aspects of brain activity. And the first one is that the brain shows rhythmic and oscillatory type of activity. It shows fast dynamics, really on sub-second scales. And the networks, so the interactions between areas, are inherently directed. Influence or interactions go from one area to another. They can also go back, of course, but the main point is there's always a push and pull. And it would be good to visualize that or model that adequately. So functional connectivity measures that can reflect these three aspects, they are closer to physiology and therefore better interpretable in terms of what the brain underneath is actually doing. So one particular measure that we've been focusing on in our work is what's called, it falls under the umbrella of what is generally known as Granger causality. So Granger causality is named after Clive Granger, but it was already developed a little bit by Norbert Wiener in the during the war. During the Second World War. Because his task was to basically track enemy planes and see where they would be at a certain point in time. And Clive Granger formalized this and made models of what's now known as Granger causality in the economic domain. So it comes from, it wasn't developed in neuroscience, but it has great use. And what Granger causality basically means is that, in neuroscientific terms, is that activity in area J, so we have here an area J, predicts activity in Y better than that I predicts its own activity. So it's a definition that is based on the predictability in time, which is close, if you think, to this directed interaction in the brain, because there's a pulse in one region happening, it goes to the other region and then modulates the activity. So activity here is predictive of what's going to happen in the other area at some later point in time. That is the basic idea behind Granger causality. Of course, our task as neuroscientists, cognitive neuroscientists, is to derive these directed interactions from our MEG or LFP or fMRI signals. And the way that's one way of doing this is by way of multivariate autoregressive modeling, or MVAR. This can estimate the predictability of a region Y from J. So essentially, what you do is you make a model of how activity at one point in time predicts future activity for each of these time series. You do this in a multivariate way. So you model them all together, and from that you can calculate measures of Granger causality. One particular measure of Granger causality that we've been working with is the partial directed coherence. And the partial directed coherence estimates the unique contribution from J to Y. So as you've seen, I've plotted here three areas. So we're interested in how much influence or how much Granger causality there is going from this area to that area. But if we want to do that adequately, we must take into account that there's also, say, indirect influences from this area on that area and then onto that area. This is why we use multivariate models. The partial directed coherence can be derived through a normalization of the MVAR model. I've listed here one type of normalization. There are several in the literature. This is the squared PDC, I think now usually we use informational PDC, but there are different types of normalizations. That's a bit of detail, but the most important thing to remember from the partial directed coherence is that we end up with a connectivity matrix that is shown here on the lower right. In this, so there's a few things to note on this connectivity matrix. First of all, it is a connectivity matrix in the frequency domain. So for each J, the area that drives, or the area that Granger causes, there's plotted in the frequency domain the amount of Granger causality or PDC that it has on the receiving area. What is the second thing is, so the first thing is the frequency domain. The second one is, this is an asymmetric matrix. So that means it is a directed measure of functional connectivity. What does that mean? Well, it means that the interactions going from from J1 to Y2 are not the same as the ones going from J2 to I1. So the other way around, basically. So those are the two key aspects of it. So partial directed coherence is a measure in the frequency domain that shows directed interactions. Well, what about the dynamics? Well, the nice thing is that we can get those dynamics when we use time varying multivariate autoregressive modeling. And the details of that I don't go into here, but they will be fully explained by David in his lecture, where he will also explain how you can take connectomes, so structural connectivity, into account as priors. And in the afternoon, this will also be the topic of the tutorial given by Maria. Good, so that all sounds very good, but as always, there are a few things to keep in mind, or limitations on functional connectivity using MEG and EEG. I think you've already heard about the problem of volume conduction, or the effect of volume conduction in EEG. And so this is a specific problem for functional connectivity and analysis, because if we have a source here in the brain, we register activity of this source at multiple electrodes at the same time. So that if you, for example, look at the correlation, and this is the correlations here, the correlation as a function of magnetometer distance, so these are MEG data, you see that even at rather large distances, correlations can still be substantial. And here's a topographical plot of that. So that means, at least when we work in sensor space, functional connectivity between pairs cannot be attributed to the underlying brain region, because you're basically looking at similarities that reflect one source. One possible solution for this, you may thought of this already yourself. You can use non-instantaneous measures or lagged measures, and Granger causality is one of those. Or you can use something called the imaginary coherence, but it's something to keep in mind. Well, if we cannot work on the sensor level, let's go to the source level, as we've seen in Peter's talk yesterday. Well, the source level will not solve your problem entirely, because the spatial resolution of inverse solutions is imperfect and inhomogeneous. So that means that your source localizations are not as accurate as we would like. They are good, you can work with them, but they're perhaps not as good as we would wish. And more important, in some regions, they're really not so good. So, for example, the deep regions pose often a problem. Here you see, for example, the correlations among activity in voxels here, in source points. So these brain areas with low spatial resolution artificially have higher functional connectivity values. Well, a possible solution of this is, of course, use a different imaging method, intracranial recordings. But this is not possible at the coverage that you have with MEG or EEG. So the main message and what is important to do is to always inspect your data. If you do a functional connectivity analysis, plot your results, see how they match with the EEG source localization. Obviously, if you see patterns like this, you must be very suspect that this is an artifact. So when we make models using network models using functional connectivity, we have to take great care, first of all, that we have the good signal and that we're aware of some artifacts. And I've only listed a few here. If you want to work with functional connectivity, please check out the references that I added in the end also, because there's always more technical things to be aware of when you really start interpreting your data. And then even when we make those models, we have to think about what is the meaning of those models. And I like to quote something famous from George Box, a famous statistician, and he's very memorably said, all models are wrong, but some are useful. And what does it mean all models are wrong? Obviously, if you have artifacts in your data, your model is wrong. But what he meant is that models are wrong in the sense that they never fully reflect the reality underneath. But they abstract certain aspects that you find useful for your theories, basically. So in this sense, there is no model that accurately reflects all of the reality underneath. It abstracts certain aspects that informs your theories and creates new hypotheses about how, in our case, the brain may be working. So that is what we try to do a little bit when we work with these time varying dynamic functional connectivity models. And to wrap it up, I'd like to briefly show you a few applications, of course, selected from work I was involved in. This is a study from David, and he showed where we did a selective attention task. So in a selective attention task, subjects get presented the same visual stimulus, and at sometimes the stimulus is relevant for them, and another time they have to just ignore it, basically. And and this is a plot of the statistically significant differences in the time and frequency domain of the functional connectivity, basically. We saw that when a stimulus is relevant, this yields an increase in directed interactions in the gamma band, whereas when they were irrelevant, this yielded an increase in the alpha band. So, and we could link this to a suppressive effect of alpha band interactions that you see here involving parietal areas. And there's some more ideas about possible mechanisms of this suppression in the paper, in case you're interested. Of course, we must also apply these methods to better understand pathologies. One example that we did together with Serge Vermier is epilepsy. And here we could show that basically using a network approach that around epileptic spikes, so these small pathological fast transient activities, that we could localize the main source of that in the left temporal lobe as predicted. And what's more, we could relate the strength of the directed interactions and where they went in the brain to the cognitive deficits that the patients suffered. This is work from Anna Cuito. Lastly and briefly, we've also applied these network measures to better understand the development of autism in children. This work by Holger Sperden was also done in Geneva. And he showed that certain interactions within what's known as the social network in the brain are already altered in autistic children of a very young age. So in sum, few things to take away from this focus. Granger causality based on EMBER models can reflect key aspects of neural interactions, particularly the rhythmic oscillatory aspects, their dynamics, so very fast transients, non-stationarities, as well as the directionality, so which area is driving and which area is receiving. There are some limitations to take into account, particularly I've mentioned two, volume conduction and field spread. and I've shown you a few applications, but really this is a rather new field that we're able to do really this dynamic directed frequency resolved modeling. So the applications are also for you to imagine. I mean, think about what you can do with your data, or where it is maybe useful as a model to inform your theories of brain function. Here are 4 references of these are just review articles to get you started if you're interested in some of the background and discussions that have been going on in the last, well, say 10 years, I think, yeah. With that, I thank you for your attention. So we'll do a few questions now, but I'd also like to say I'll be in the discussion room during the break. So if you just want to chat or discuss a little bit more informally, I hope to see you there. Thank you very much.